So good morning, everyone, and thanks for having selected my abstract for this short talk. So I'm Tiziana La Bella, and I work at Genethon in Giuseppe Ronzitti's team. And today I'm going to talk about the rational shuffling, a novel engineering method, a novel capsid engineering method for adeno-associated viral vector. So first of all, the adeno-associated viral vector are derived from a natural virus, which is known to be replication defective. It's composed by a small genome of 4.7 kilobase. It's a single-stranded DNA genome that is encapsulated in a non-enveloped capsid. So the virus is very small. It's around 20 nanometer. And is known to naturally infect human, but is also considered mostly as non-pathogenic. Concerning the vector, what we found is that we still keep the presence of the viral capsid, whereas for what it concerns the vector genome, all the viral coding sequence are depleted and replaced by the expression cassette. One of the major advantages of the AV vector is that they are non-integrative, and they predominantly persist as a transcriptionally active form, allowing for the long-term expression in postmitotic cell. Currently, the AV vector are the leading platform for in vivo gene transfer, and one of the major advantages of using this vector is that they can be applied for a broad spectrum of disorder. Here in the pie chart, you can see an example of the number of clinical trials for different disorder. And all the increasing number of clinical trials using AV vector support both the safety and efficacy of this delivery system. And this led in particular to the approval of 7 AV-based drugs which are currently on market. Concerning the capsid that we use in the clinical setting, we mostly found natural variant. However, in the most recent clinical trial, we start to see also the usage of engineered capsid. And this is particularly important because capsid engineering can help to overcome one of the issues that we have with natural capsid, which is the broad biodistribution. So by engineering a capsid, we can make this vector more specific to target a specific organ or cell type. Another issue related to the use of wild-type capsid is the fact that since the virus naturally infects human, we have the presence of neutralizing antibody against the capsid. And in particular, this is an exclusion criteria for 45% of clinical trial. And when we look about blood disorder, which are on the bottom of the graph, you can see that this percentage of exclusion reached the 90%. Also in this case, capsid engineering can help to find an alternative to bypass this issue related to pre-existing immunity, for example, by mutating the epitope that are recognized by the antibody. So concerning our project, we started by isolate novel capsid variant from human. We characterize them in terms of production, in vitro and in vivo biodistribution, and seroprevalence in human. And we selected some of those variants. Then we use this variant. There is an issue with the presentation, but that's fine. Then we use this variant to make new capsids, so to engineer novel capsid. And finally, of course, the last step will be the validation in non-human primate. And I just wanted to precise that all the data that I'm going to show concern vectors that are produced using a luciferase as a reporter gene under the control of an ubiquitous promoter. So here in this phylogenetic tree, you can see the 56 variants that we isolated from human liver. And in particular, the variant cluster in two branches, one which is very close to the AV2 serotype, and the other one which is an hybrid between an AV2 and an AV13. So we started by produce those variants, and we found that the 70% of them were successfully produced, and all the produced variants were able at least to transduce cell in vitro. So we moved to the biodistribution following intravenous injection in mice. And as you can see here in the heat map, the majority of the variant had this marked tropism for heart, for all the muscle, and for central nervous system. And we also observed this very low transduction efficiency in the liver. The issue when we use this wild-type variant is, of course, the fact that they are very seroprevalent in human. And here you can see an example of one wild-type capsid where we found that the seroprevalence was comparable to the one of AV2. And just for people that are not in the field, AV2 is the most seroprevalent capsid in human. So we wanted to find a way to be able to use these interesting biodistribution properties of the wild-type capsid, but avoiding this issue related to the seroprevalence. And we developed this method called rational shuffling, which aim to transfer the biodistribution properties from a donor capsid, so a wild-type capsid in our case, to an acceptor capsid that is a capsid with low seroprevalence. And the goal is to avoid to modify the seroprevalence and the productivity of the acceptor capsid. So to transfer those properties, we focused on the hypervariable region, which are represented here with different color in the image. So the hypervariable region are the looped out domain of the viral capsid protein. So they are localized on the surface of the capsid, and they are known to interact with the host cell receptor. So we hypothesized that by transferring this hypervariable region, we could transfer the biodistribution properties from the donor to the acceptor capsid. And this is exactly what we tried to do. So here you can see the result of the first 5 mutants that we designed, where we transferred a decreasing number of hypervariable region. And concerning the production, you can see that the yield was totally comparable to the one of the parental capsid, which in this case was an AV8 as acceptor capsid and one of our wild-type capsid as donor capsid. For the biodistribution, we observed that 4 out of 5 mutants had this marked tropism for heart and the different muscle and the central nervous system, and also this low transduction efficiency in the liver, which is exactly the profile that we found in our wild-type variant. And interestingly, concerning the seroprevalence, we observed that the mutant 3, 4, and 5 had a level of neutralizing antibody that was comparable to the one of the acceptor capsid, which is exactly what we wanted. And for the mutant 1 and 2, we observed this increased level of seroprevalence, but it was expected because we transferred a higher number of hypervariable region. So we conclude by saying that we could really use this method to transfer the biodistribution properties without altering the seroprevalence of the acceptor capsid and the production also. So starting from that, we decided to make a collection of rationally shuffled capsid in which we use 3 different capsids as acceptor, which are all capsids already used in clinic, and all our wild-type capsid as donor capsid. And we make this library of 86 engineered variant. We characterize them. So we found first that the 88% were viable, which is a very high percentage. And then we characterize them. And here in this heat map, you can see the global result of the biodistribution that we had for all our variant. And again, you can see this tropism that is increased for heart, for the different muscle, and for the central nervous system. And concerning the liver, we still observe this globally low transduction efficiency. So just to conclude, we isolated these 56 novel capsid variants from human. We characterized them, finding mostly these marked tropism for muscle and for central nervous system. And we use this variant to develop a novel capsid engineering method, which allows us to transfer the biodistribution properties from a capsid with high seroprevalence to a backbone capsid with low seroprevalence. And of course, now the final step will be the cross-species validation in non-human primate to confirm the translational potential of those capsid, and also to better characterize the hypervariable region that are responsible for a specific tropism. And with that, I conclude my presentation, and I want just to thank all my team, in particular my boss, Giuseppe Ronzitti, and all the people that work with me in the gene therapy technology group within the team, and all the foundings, of course, and thank you all for your attention.
Any question? 200 bright minds here that need to wake up. Any questions? Okay, so I'll try my best. I guess you don't have enough data, but what kind of data would you need in order to be completely predictive in it? So now you did a sort of a rational, rational shuffling between existing variants. Does this give you enough information to say, okay, now I can try to predict even a better variant or a more specific one for specific applications?
We still miss something, is that we are transferring a whole hypervariable region from the wild-type capsid, but we don't know exactly which amino acid, which residue of the hypervariable region are responsible of this tropism. So what we want now to do, what we are doing actually, is to focus on the most interesting hypervariable region and doing a library of mutation in the specific one. And then like that we'll be more able to fine-tune really the design of our capsid for a specific application.
Sounds like a good project for a biofoundry. Thank you very much.
Thank you.